Hello, everyone. Thanks for joining us for a special look at what our restoration teams are doing on a ship in the deep Gulf. Hi there. My name is Sasha Francis. I'm the Gulf Restoration Education and Outreach Manager at National Marine Sanctuary Foundation, and I will be your host today. So we'll get to talk to our friends on a ship named the RV Point Sur, which is a 135-foot vessel owned by the University's Southern Mississippi and operated by the Louisiana University's Marine Consortium, or LUMCON. Today, the team is currently about 100 miles off the coast of Mississippi. So throughout today's program, please share any questions or comments that come up in the chat. We'll pause for questions as we go. All right. Also, we'd love to get your feedback on today's program. So we have a short survey you can fill out to tell us a little bit about your experience. We'll send the link to the chat now and again at the end of the program. We would love to have you fill that out before you log off today. So in just a few minutes, we'll connect with the folks on board. But first, we're going to talk about why they're out there and what they are up to. So we're going to start with a little bit of overview here. So as you travel deeper in the ocean, less and less sunlight is able to penetrate the water. Just past recreational scuba diving limits, you reach the mesophotic zone, meaning middle light. Despite the low levels of sunlight, this zone is home to colorful corals, sponges, fish, and invertebrates. And beyond this twilight zone, there's no sunlight at all, as well as colder temperatures and higher pressure. But there are still vibrant habitats full of life at those depths, hundreds to thousands of feet below the surface. These mesophotic and deep benthic, or seafloor ecosystems, are found worldwide, including in the Gulf, which is where all this beautiful footage is actually from. However, the 2010 Deepwater Horizon oil spill dealt a heavy blow to Gulf communities on land and at sea. 11 people lost their lives, and natural resources, wildlife, and habitats across the northern Gulf were severely impacted. Over 770 square miles of deep sea habitat were impacted by the spill. As a result, collaborative restoration projects are underway to help us understand and restore these seafloor ecosystems found below 160 feet deep. These projects are developing new and improved maps of the seafloor to locate these important coral communities. They're learning more about the species that live in these habitats, the environmental conditions they thrive in, and how they may be connected across the Gulf. Determining the best methods to encourage deep sea coral growth and reproduction to help restore impacted coral species and the important habitat they create. And also reducing the threats that these ecosystems face. And connecting the habitats and restoration work to the public and natural resource managers through programs like this. This restoration work is made possible by the Deepwater Horizon Natural Resource Damage Assessment Open Ocean Trustees and is led by the National Oceanic and Atmospheric Administration, or NOAA, and the Department of the Interior, or DOI. Plus many partners, including a large number of academic institutions and non-governmental organizations. All right. So now the exciting part, and do feel free to save any links we put in the chat today about more information about our projects. We'll be including those as we go. So now the fun part, we're going to get to chat with some folks on board. They're at sea right now doing some very cool science. Hello. Hi, friends. How are you today?
We're good.
I love the octopus microphone cover. It's adorable. All right. Do you want to introduce yourselves and tell us what you're up to on this mission? What are your main roles and where you work?
Hi, I'm Jill Burke. I'm a benthic ecologist with the U.S. Geological Survey based in Gainesville, Florida. I am acting as a field party chief for this mission and generally the sediment lead for the mesophotic and deep benthic communities restoration project.
My name is Christia Bush. I'm based out of Leetown, West Virginia, also with the United States Geological Survey. I'm part of the population species dynamics group, and I'm here on board collecting water samples for environmental DNA studies.
Awesome. And we're going to talk more about that. And so folks that might not be familiar with USGS, I'm sure many people listening have heard about USGS. They are part of the Department of Interior. So when I mentioned that this work is led by NOAA and the Department of Interior, that includes lots of hard work by USGS folks just like you. So thank you for all that you do. So I wanted to ask you a little bit about kind of your work over time out here in the Gulf, particularly as it connects to this restoration work. So this mission is part of a series that's taken place over multiple years, multiple seasons. So what are you hoping to accomplish on this mission?
So, as Sasha mentioned, the Deepwater Horizon oil spill resulted in very large areas of the seafloor being injured. This included soft sediment habitats, or the mud habitats, which is what we're focused on out here. Estimates from work done shortly in the few years after the spill suggested that recovery in these habitats could take decades or longer. So, our first question that we wanted to look at was get an updated status of how are these communities recovering? What are they looking like a decade later? And so we first started looking at interannual variability, see how they're changing both in impacted and non-impacted areas. But now the cruise we're on right now is a series of 4 where we're looking at seasonal variability to give us a better idea of how those fluctuations change in the Gulf across these both injured and natural habitats.
Pretty much covered it.
Awesome. And so have either or both of you been out at different times of year working on this particular series of missions? And what have you seen? How do we know like how the mud environment changes across the different seasons, or the water sample for water sample data, for example?
So far we've mostly been out in September and April, so getting a spring and a fall time point. But we suspect that there's increased productivity in surface waters, which is what fuels a lot of the biomass and density of animals on the seafloor. All that food gets down to the bottom and feeds things. And so changes in these factors can influence the communities both on very short time scales to much longer time scales based on the life history of the animals living there.
Awesome. What's the value of collecting this type of data at different times of year and across multiple years? How does this inform the restoration work that we're doing?
Yeah, so as we progress like from the event, the oil spill, there's obviously recovery periods, and the oil can still impact the communities even like years after. And so getting an idea of how everything is changing as we're progressing year to year, we're able to make comparisons, seeing how, so specifically like with what I'm looking at, we're able to take the water samples, and specifically with eDNA, that's environmental DNA. So cells are released off of organisms, and it's in the water column, and we're able to take the water from different levels and depths within the water column and process them in the lab, and we're able to identify what species the different cells are coming off of. And so we're able to, take that and get an idea of the diversity in the different areas that we're sampling from, and identify, how it changes from year to year. And then also, depending on like temperature changes, like season by season, there's obviously differences in the organisms that are in particular areas. Fish will migrate in and out of the Gulf or to different areas within the Gulf, as well as different organisms. And so we're able to, by looking at season by season, we're able to see kind of how they're moving around and compare, how those are changing throughout the years of this project.
Another thing for the sediment communities in particular, by understanding this natural, both short and long-term variability, we're able to put context to both the legacy data that may have been collected at different times of the year, and also future data collection. We don't always have the luxury of choosing when we can go to sea and when we can collect samples. It helps provide us that understanding for future monitoring and assessment.
Yeah, that's a really great point too, because you can't always perfectly pick what time of year you're going to go. And then even when you do, sometimes things change, right? So can you speak a little bit to what it's like working the Gulf just across? I mean, we're out there usually March through October. And anybody who lives down here in the Gulf, or even those if like Cassia living on the eastern, the North Atlantic area, about some changes that come through. So what's it like going out to sea and kind of, I guess, having to stay on your toes and kind of be flexible during this season? Can you speak to that a little bit?
So yeah, a little bit. I mean, especially September, we've been out multiple years. We're right in the height of hurricane season, and we have been essentially chased into shore by hurricanes. And so you try and prioritize the sampling you need to get done and your mission objectives to try and get everything in, but still keep everyone safe. So it is always a juggling act. Luckily this year, we have had fantastic weather. We've been able to work every day and collect so many samples. It's been really exciting.
Yeah. And I live in Galveston, Texas, so very familiar with those changes and needing to stay on your toes for hurricane season. I'm really glad y'all are having great weather though. So I guess that's a really important note about how great it is to have long-term data, right? Because maybe you really want to collect every April or every September, but sometimes those windows might change and it might not be up to you if you just can't get out to sea because of bad weather or because of other issues that pop up. So it's really amazing to be able to have that long-term data over time and be able to compare as best as you can. So I wanted to talk a little bit more about eDNA because I know there's probably a lot of people watching that are like, what is that? And you mentioned it's environmental DNA, but really kind of you mentioned that it helps us kind of understand what all is down there. So how can that help us restore these habitats better?
Yeah, so eDNA is a up and coming field right now, and it's actually very beneficial in its ability to be non-invasive sampling. And so typically to get ideas of what fish or organisms are present, you would have to either get a physical sample or you would have to get video footage and then be able to spend hours combing through this video footage and identifying, what organisms are seen or, identifying in the physical samples that are brought up. And eDNA is amazing in the fact that it is a much simpler process. Well, it's not simpler, but it doesn't require all those extra hours of identification. You're able to just, collect water sample and you filter it through, and by extracting the DNA and testing it against different markers which identify what species are present in the water, you're able to get an idea of, throughout the water column, like what species are there. And it doesn't quite tell us amounts or the density of a particular organism, but it can give you an idea of like the number of organisms that are present. And who knows, as technology progresses and advances, potentially at some point in the future, it could help to get population levels, but it's not there. It's not advanced enough at that point yet.
So when you take the DNA sample, oh, sorry, go ahead, Jill. Do you have something to add?
I was just going to say it also helps us get at the connectivity among our different habitats by seeing what animals are located in multiple different places and how they might be interacting between areas.
That's a really good note too. We really can't restore these habitats until we fully understand them, right? And these areas are not areas that have previously been really well understood. So a lot of the work that we're doing in these restoration projects is really getting a better understanding of these habitats through higher resolution seafloor maps that we have before we go into a space, and use the ROV to go down and get a really good idea of what it looks like down there, the different characteristics of the seafloor, understanding where these different coral communities and other species might be living down there, and then using the sediment and the water samples to really get a really good picture of what lives down there, what relies on these coral communities to survive and to thrive. And so, yeah, that's a really good point, is all the connectivity of it, so that we can best know ideally what these habitats should look like and what all, relies on each other across these different communities. So I had a question about the eDNA. So when you basically say you find, evidence of DNA in the water, and then do you have to personally like compare each DNA reading that you get across to compare it, or is there like, can you run it through a system to do a match? I'm just curious how that process works.
Yeah. So I'll kind of walk you through the whole process. So we collect the water samples and filter it through, and then we take the filter and we're able to extract the DNA by breaking apart the cells and put the DNA into solution. And then we have different markers, and so those are specific to different groups of organisms, like fish or invertebrates, um, bacteria, stuff like that. And then once each DNA is marked with those particular markers, we sequence it, which just gets pretty much your DNA read out as like a code of letters. And that is compared against a baseline. So we have a pipeline, and it's basically known sequences of DNA from different organisms. And by comparing the samples that we have, which are unknowns, to these knowns, and the computer does all of those comparisons, we're able to identify what species are present. So yes, there is a pipeline that we send all of these sequences through, and it is what identifies out to the species level, or sometimes we aren't able to get specific species levels and we're higher, we get like genus levels or family. We try to get as specific as possible, but sometimes depending on how degraded the DNA might be, we may not get all the way down to species level.
So if you see something that like doesn't look familiar to you, for example, is it possible that it's a new species that maybe you could detect through eDNA?
It has happened before. I think we have come across new species during this particular project. And so, yes, if you see something and it has a diversity that's pretty different than anything that, maybe similar to it, then, you can't necessarily identify that it is a new species through eDNA, because you would have to identify and find the physical item, and then you would genotype that particular item. And if it has the same genotype as the unknown sample, then you could identify a new species. But I think we have identified some new species out on this project.
Very cool. Awesome. Jill, did you have anything to add to that?
I was just going to say, so they've been doing eDNA processing for water samples for a long time, but we hope to have goals of processing sediment as well. So new on this cruise, we've been collecting individuals from the sediments and isolate them to ultimately add to these genetic libraries that the eDNA gets compared to. So help inform future efforts with these are taxa we know are found, and if we detect them throughout the Gulf.
Awesome. All right, let's see. I just got word that it seems like YouTube is having issues and our chat is currently disabled. So we have not had anybody be able to put in questions. I'm so sorry if you're watching and you have a question. We're going to try to find a solution for that. Okay. It looks like now our folks working behind the scenes have figured that out. So if you did have a question and you didn't get to type it in the chat, please try again. If you need to refresh or anything, that might work, but we'd love to answer your questions if you do have them. So sorry about that. All right. So another question I have. So how do factors like the Mississippi River, kind of the watershed system, and rainfall across different seasons contribute to changes we might see in the samples that you're looking at or in the Gulf as a whole?
So that's part of why we suspect there is seasonality in the Gulf, is the increased river outflow in the springtime from the Mississippi River, which outputs extra nutrients, extra sediment out onto the shelf and slope areas in the Gulf. So that ultimately should lead to increased surface productivity, so increases in phytoplankton, zooplankton, which ultimately counts as food as it slowly makes its way to the seafloor. Of course, the question then becomes, what sort of lag is there in seeing a response in that increase in food availability in our communities? We have our microbial communities, which would likely respond very, very quickly to any changes. Our myofauna community is a little bit longer, and then our macrofauna the longest, because just life history, reproduction, how long it takes for a population to increase or decrease based on those factors.
Yeah, I think we talked a little bit more deeply about kind of those different communities on our last live stream, which was like Creatures of the Mud that I did last week. So if anybody watching is interested in more kind of getting in depth with the scientists that focus on those different kind of sizes and microbial communities, check out that live stream. We have some more USGS folks and some more specialists talking about that too. And living in the Gulf too, especially in Galveston here, we get a lot of people that are like, your water's all dirty and it's so brown, and why is it so brown? But I think, talking about that inflow of sediment from the Mississippi and from the watershed, it's not a bad thing, right? It can definitely contribute to kind of mixing things up and creating a new nutrient flow and having that natural seasonal change. Obviously, right? If there's too much rain or too much runoff or other things, human runoff coming into the watershed through, pollution or, things like that, then that can contribute to changes, I imagine, as well. All right, let's see if we have any questions yet from that. Not yet. Okay. All right. So I wanted to ask another question before we wrap up and see if we have anybody else chiming in with some questions here. But in addition to the mud samples and the water samples, the restoration teams are collecting all kinds of other samples and data across the Gulf. How can all this information kind of be pieced together to tell the broader story of the Gulf ecosystem? So what are some other samples that we're collecting out there?
So yeah, one of the beauties of this project is it is very multifaceted and trying to get at that whole ecosystem picture of what's going on. So besides the sediments and the eDNA, we're taking water column measurements themselves with nutrients to see how those change throughout the water column. Not just sediments like that we're collecting, but we also collect sediments near coral habitats, which have been shown to be different communities than the sediments just when you're just out in the regular mud. So how those different habitats are interplaying to influence each other, to get at that broader picture, to ultimately inform where we are doing active restoration and inform those decisions.
Yeah.
We also have, sorry.
Go ahead.
I was going to say some of the other data that we've been collecting is bottom mapping, and so that's beneficial. We're able to get an idea of the bathymetry of the bottom mounds that may be habitat for corals versus deeper areas, and so that definitely plays a part in as well in the entire ecosystem. And so finding how the bottom, the mapping, merges together with what populations are there, which we can get from video footage from like ROVs, which is another portion of this project as well as the DNA samples and physical collections from trawls, or the ROVs are also able to collect physical samples. So yeah, it's amazing how everything is able to work together And everybody has, their own like niche topics, but it all blends together really well to get a great idea of what's happening here in the Gulf.
Yeah, definitely. And for folks that want to participate in helping us, characterize the Gulf and characterize the seafloor, or see what's down there and identify what's down there, there is a really cool community science project called Click a Coral, and that people can actually watch the video footage from the ROVs and actually be able to help us identify the different species we see down there. So corals and sponges and fish and vertebrates and things like that. So we'll try to grab that link so we can put it in the chat. So yeah, you can see some footage on screen right now of the ROV working to explore some of these coral habitats so we know exactly what's down there so we can best restore it. And also, like they mentioned, be able to collect some coral samples. And so another thing to mention is we have coral labs where we're actually taking care of live corals and supporting their reproduction so we can learn more about how they reproduce and what they might need to reproduce better so we can help make up for some of those colonies that were lost from the oil spill. So we actually are doing some outplanting with corals as well. And the sediment and water sampling and eDNA work really helps us best understand how everything is all connected and where we might put these coral racks out to help them grow and really do the best they can, have everything they need. Awesome. Well, I'm going to just double check right now to make sure we don't have any questions. We have a hi mud friends from Layla. One of our other scientists joining in the chat to say hello to y'all. Awesome. So I just wanted to end with a kind of fun question. So I know some folks out there might not be very fascinated by mud or water sampling, and I hope they have a better appreciation for it now that y'all have told us how much it helps us really understand the Gulf ecosystem and how much it helps us best restore these habitats that were injured by the oil spill. But can you tell me why you kind of got connected to this work, like why it matters to you, why you get excited to participate in this restoration?
Yeah, I mean, so growing up, I was in Ohio, no ocean at all. And I've just really come to love the ocean through like my vacations and stuff. And that's really the only experience that I had. And so I knew that I wanted to get into marine work. And honestly, it's been amazing to be able to have this job. And I look forward to it every single day. And just knowing that, we're able to take an unfortunate situation such as oil spills but use it to gain knowledge and gain a better understanding of how everything in the Gulf and ultimately in all of our oceans work and how all of these little niche topics will blend together. It's just fascinating.
So I probably fell in love with the invertebrates is where it started. There's just so many different tiny animals that live in the mud that are just so fascinating. You never know what you're going to see when you look in a sediment sample. But for this project, I started with USGS right during the original response from the oil spill. So it's been really great to come full circle to now the restoration phase to see how things have changed and where we're going and all the different things we've been able to learn about the Gulf since then. It's really great.
That's amazing that you've, been able to see kind of the worst and now see what we're doing to help. And so I'm sure that gives you some hope, right? To see everybody really working together to really try to do the best we can to help these habitats, especially when you see like a really cute tiny creature that probably people don't even know exists, right? Because it's just in the mud. Incredible. So thank you all so much for your time today. What I really love about these projects and working on these projects is getting to learn so much from all the different specialists like y'all. When I jumped into this project, I really didn't know much about mud, sediment sampling, water sampling, and I really think it's so cool now that I've gotten to learn from especially USGS folks about all the important things we find in there. So thank you for sharing your knowledge with everyone. And we appreciate you taking the time today. Have an ice cream for me on board. and hope y'all continue to have great weather. We'll let you get
Thank you
back to
so
work
much.
now, and we'll wrap up here with some more links in our chat. Thank you all for joining us. All right. I don't think we have any more questions in the chat. So sorry again for everybody if you were trying to put questions in the chat and we had that disabled for some reason today. But if you like today's event, let your folks know, tell your friends and family, check out upcoming events from the University of Rhode Island's Inner Space Center YouTube page. You can subscribe to that page so that you know when new live streams are coming up or new videos are added. You can watch recordings of our past live streams and find future live stream events on that page. And they're going to drop a link in the chat to make sure you know where to go. And to learn more about these restoration projects, find articles, publications, our interactive story map to see where our expeditions have gone in the past, and hear about updates, check out the other link provided in the chat that says learn more about deep sea restoration in the Gulf. And again, we'd love to get your feedback on today's program. We mentioned it at the beginning of the program, but we're going to have it here too. Please fill out the quick survey that you have linked in the chat if you have a moment, or you can copy and paste that link after we end today so that you can do it later today. We appreciate any feedback you have. Sorry again about the chat issue, but I'm glad we got that resolved for the end of the program. And thanks so much for joining us today, and we hope to see you next time.